Welcome, everyone. This is the inaugural seminar of the Future Leaders in Mechanobiology seminar series. And so this is a monthly seminar a series showcasing senior PhD students and postdocs from diverse backgrounds that were nominated by the mechanobiology community. So the goal here is really for these early stage researchers to share their work to a broad audience and give them an opportunity to interact with researchers at really all career stages in the mechanobiology field. So future leaders in mechanobiology will meet via Zoom every second Wednesday of the month. So stay tuned for additional announcements. So I'd like to introduce our first speaker then. Today, our speaker, Melina Gyparaki. Am I saying that right? Okay, good. She was born and raised in Athens, Greece, and received her bachelor's degree in molecular genetics from the University of Edinburgh in Scotland. During her undergrad, she gained experience in exploring microtubule dynamics in plants and yeast using several imaging technologies. When she joined Penn, University of Pennsylvania, to start her PhD, Melina quickly became interested in the use of super resolution microscopy to study cytoskeleton. After attending a talk by her now PI, Melina Gyparaki. Melina's current work is focuses on the study of microtubule-associated protein tau, which she's going to talk about today. Melina is also interested in science outreach, and a fun fact, she enjoys bar workouts. And this is not a bar where you go to get your favorite drink. This is actually a ballet bar workout. All right, so Melina, you can begin.
Thank you so much for the introduction, Trina. Can you all hear me well? Great. So hi everyone, I'm Melina and I'm a grad student in Dr. Melina Gyparaki lab at Penn, and I'm very excited to be giving a talk to you all today. Thank you so much to the committee for nominating me to present today. I'm very grateful. So today I will be talking to you a little bit about what I've been working on as part of my thesis the past few years, specifically about using super resolution microscopy to study tau aggregation. I will also be presenting a couple slides on a newer project that we're working on, but it is relevant to tau aggregation, and that will be the last few minutes of the talk. So to start off, I want to give you a brief introduction on tau, which is the protein of interest in my case. And I'm trying to move to the next slide, but it's not. Okay, there we go. All right, so tau is a microtubule-associated protein, or MAP, that's mostly found in neurons in humans. And in this schematic here, I've included, a schematic of a neuron, and you can see the microtubules running down the axon of the neuron. And if we zoom in, we can see that tau is associated with the microtubules, and it also exists in a free cytosolic form. Now, tau's function is very important for the stability of the microtubules on the axon, and having proper stability of microtubules in the axon is very important for axonal transport of proteins and also for maintaining, the overall structural integrity of neurons. Now, tau can also obtain several post-translational modifications, one of which is hyperphosphorylation. Now, in particular, tau can become hyperphosphorylated, and as a result, it can fall off the microtubules, and it can begin to aggregate, initially forming small, soluble aggregates, and later on forming much larger insoluble aggregates, such as paired helical filaments and neurofibrillary tangles, or NFTs. These NFT structures have been considered to be one of the main pathological features of Alzheimer's disease, as well as other neurodegenerative diseases that are collectively known as tauopathies. Now, for a long time, researchers had been focusing on the study of NFTs to be the main toxic tau species. And that was because they appeared basically in images of postmortem brains of patients. However, it turned out that neurodegeneration, this is something we found out from more recent data from animal models that actually neurodegeneration can precede this NFT formation stage. So everyone's attention sort of shifted to earlier stages of the tau aggregation pathway, and in particular to this soluble tau aggregate stage, which is otherwise known as the oligomeric tau aggregate stage. Now, as you can see from the scale I have used here, these oligomers are very small in size. They range between 6 to 35 nanometers, which is in fact so small that they cannot be visualized in intact cells with conventional light microscopy, which has a limit of resolution around 200 nanometers. So basically, the lack of sensitive high-resolution tools to visualize these structures is making it more challenging to determine the stoichiometry of tau molecules within these tau oligomers. So, and without knowing this information, it's sort of harder to know how to target these structures for therapeutic purposes. So in the Lacadamiali lab, we use different types of super resolution microscopy to study cellular processes. So we can get down to around 20 nanometer resolution with our approaches and are able to visualize these structures in intact cells. Or at least that was the idea before we started. So the two main goals that I had, in my project were to determine the nanoscale composition of tau on microtubules and in tau aggregates. And this first part is what I'm going to focus for most of the talk, and everything I'll present for this part has been published. It was actually just published last week. And for the second part, we are looking a little bit about how tau aggregates get cleared and which degradation pathways are important for this clearance. So before I go into goal one and talk to you about exactly what we did, I want to introduce the super resolution method that we are using for experiments. So we're using a type of single molecule localization microscopy known as STORM. So STORM stands for Stochastic Optical Reconstruction Microscopy. And to give you an idea of how STORM overcomes the limitations of conventional microscopy, I'm just going to start with conventional microscopy. So imagine that you have a sample that has been fixed and you have chose a specific protein of interest that you have stained using an antibody and maybe a secondary that has a fluorescent dye. So when you visualize your sample with a conventional light microscope, all of the fluorophores in your sample are going to be active at the same time. And that's going to lead to a problem where if you have two fluorophores that are too close to each other, below the resolution limit of conventional microscopy, so below 200 nanometers, they're going to appear as this unresolved blob on the right. Now, in order to overcome this issue, what can happen with STORM using appropriate buffers as well as photoswitchable molecules is that the molecule on the left can be put into an off state or a dark state, as we call it, while the molecule on the right can be stochastically activated and its centroid position can be localized with nanometer precision. Later in the acquisition, the molecule on the right will be put in the dark state, while the molecule on the left is stochastically activated, and so on. So eventually, you can imagine that this will happen for all the fluorescent molecules in your sample, and you will end up with a reconstructed image containing localizations or points in space stemming from all of the fluorescent molecules that you have in your sample. And that's going to be your STORM or super resolved image. So to see this in action, I've included here on the left a conventional fluorescence image of tubulin. So we're looking at microtubules. And in the middle image, you can see we see this blinking effect, which corresponds to the switching on and off of the fluorophores. And then on the right, we see how the STORM image is reconstructed. So using STORM, we wanted to tackle the question of what is the nanoscale composition of tau under what we loosely refer to as physiological versus pathological conditions. And what we really mean here is in the former case when tau is non-aggregated versus when tau is aggregated in the latter case. So we hypothesized that there's going to be a wide distribution of tau molecules on the microtubules in physiological conditions. And to test this hypothesis, we used a cell model that we obtained from Virginia Lee's lab here at Penn, and I've included here the reference where this cell model first appeared. So this cell model is engineered to contain a construct of tau that has GFP and a mutation in tau called P3ONL. This is a disease-relevant mutation. And the construct is DOX-inducible, so it can only be expressed when you add doxycycline in the media. And it consists of two clones, clone 4 and clone 4.1. The presence of the mutation itself in this cell model is not sufficient to cause tau aggregation. So clone 4 looks like the tau looks microtubule associated. So this is our physiological conditions. Whereas clone 4.1 is the sorted clone of clone 4 that has been created after the exogenous transduction of tau fibrils and propagated in culture. So tau looks very aggregated. So we have this system of non-aggregated versus aggregated tau. So the first thing we wanted to look at was basically how does the tau look on the microtubules. So we took clone 4, where tau is non-aggregated, and we performed storm imaging. So I'm showing you here a storm image. So a reminder here, our imaging is density-based, so we get points in space that we can render by giving them a blur for visualization. So that's what we see here, the rendered image. And I have zoomed into a specific region of this image, and in the bottom zoomed in set, I'm also showing you how this image is segmented. I have stained the tau using a GFP nanobody that contains a photoswitchable dye. So that's what we're looking at. So when we're looking at this image, we can see that P3ONL tau is not uniformly distributed on the microtubules, as in it does not fully occupy the microtubule polymer, but instead it forms what we call nanoclusters. So what are nanoclusters? Nanoclusters are groups of localizations that correspond to fluorescent molecules in the sample. So on the image on the right, you can see here the different pseudocolors representing different segmented nanoclusters. We don't have enough colors for all the different nanoclusters in this software, but you can distinguish the different nanoclusters by looking at the white dot that's found in the center of the nanocluster. So this, like, I don't know if you can see my pointer, but this blue nanocluster is different from this green one, from this red one, and so on. And different nanoclusters can have different number of localizations. So it was interesting that we saw this formation of nanoclusters, and we wanted to make sure we're looking at tau that's on the microtubules at this point. So the next thing we did was a two-color storm experiment where we stained with for both alpha tubulin and GFP tau using the GFP nanobody we used previously, and performed a colocalization analysis. We actually developed our own colocalization algorithm for this. And what this algorithm does is basically it will take the alpha tubulin images as a mask, and it will ask how much of tau falls into the tubulin mask. And for that result, you can see here on the right, whatever colocalizes, like the colocalized clusters, are going to be in magenta, and whatever does not colocalize is going to go into the isolated clusters in yellow. So we figured out that our colocalization algorithm was actually underestimating true colocalization. So after performing a control experiment, we corrected for this underestimation. And with the corrected data, we found that we approximately like 100% of the tau nanoclusters were microtubule associated. So it gave us confidence that tau is definitely on the microtubules. So the next thing we wanted to see was, what is the nature of tau within these nanoclusters we observed? Is it monomeric or oligomeric? And to answer this question, we used several different approaches, but the first one was pretty straightforward. We obtained an oligomeric tau antibody that's commercially available and stained the clone 4 non-aggregated tau cells with it. And as you can see from this storm image I've included here, the staining of this T22 antibody reveals a microtubule-like pattern, suggesting that, this antibody binds to microtubule-associated tau. And as you can see from the segmentation I've included here, we do see the formation of the nanoclusters with the staining of this antibody as well. So then, again, we performed a two-color storm experiment looking at both T22, so oligomeric tau, and GFP tau. And after performing the colocalization analysis, we found that we have approximately 34% of tau nanoclusters colocalizing with T22 stained oligomers. So at this point, we were more confident that there is a population of microtubule-associated tau that is also oligomeric. So the next step here was, to determine the copy number of tau within these oligomers. And for this part, we actually used an approach we have previously used to estimate the copy number estimate basically the stoichiometry of proteins in storm super resolution images. And to do that, we use monomeric protein images as a calibration. So you're probably wondering, where did you get the monomeric tau images for this part? So for the calibration to obtain the monomeric tau images, I performed a dilution experiment. So for all the imaging I have showed you so far using the GFP nanobody, I have been using a regular dilution of the nanobody of 1 to 100, which can fully saturate the epitopes present. So now I did an 100-fold dilution down to 1 to 10,000 to very sparsely label the sample. So as you can see on the plot in the left, where we're looking at the distribution of number of localizations per cluster, we have a significant drop in the localizations per cluster for this diluted sample. And if we look at the storm image I've included here on the right, we will see indeed that the labeling of tau molecules is very sparse. And basically, based on these data, we assume that the resulting nanoclusters that we get in this 100-fold diluted sample corresponded to monomeric data. And the reason why we still have nanoclusters present in the diluted sample could be due to repeated blinking of the same molecule. Now, this is a common artifact that can happen in storm to get nanoclusters forming from repeated blinking of the same molecule. And before you ask me if that's also the case in the previous data I showed you so far, I will say that we tested for it. We used an algorithm that's called the Ddc algorithm. It was developed from Jia Xiao's lab at Johns Hopkins. We collaborated with them and they ran the algorithm for all of our data. And we saw that even after correcting for repeated blinking, we still had nanoclusters. Whereas when we corrected, in this case, for the diluted sample, we didn't have nanoclusters. So we are confident that we're looking at monomeric tau that we can use as the calibration for to estimate, the copy number that I alluded to earlier. So for determining this copy number, the next step was to take the data, the number of localizations per cluster data, that's our calibration. We fit it to a log normal distribution function, and then we performed a fitting using our actual experimental data of the regular dilution, and it showed us that we approximately have 60% monomers, 20% dimers, and 20% trimers in our samples. So a 60-40 monomer oligomer ratio. And here I'm reminding you that when we did our colocalization analysis with the oligomeric tau antibody, we found approximately 34% of colocalized clusters that were oligomeric, which is pretty similar to the 40% we found from this fitting. So these data convince us more that we have a population of tau that is both microtubule associated and oligomeric. In case I haven't convinced you so far, I also included some other control experiments that we performed to exclude some other possibilities that could be behind the formation of nanoclusters. So our first concern was whether the mutation that's in the construct of the cell model could be causing this nanocluster formation. So to exclude this possibility, I looked at mammalian cell lines that we have in the lab that contain a different tau construct that has GFP wild-type tau. I actually looked at more than one construct. I've included one here for space limitations. So when we looked at GFP wild-type tau lines, again, we saw that there was nanocluster formation. Then we wondered, could it be GFP that's causing nanoclusters? So again, to exclude that possibility, I looked at neurons that endogenously express tau. And I stand here with the tau-5 antibody, which is a total tau antibody, and we saw that there were nanoclusters forming. I also stained neurons with the T22 antibody, which specifically stains oligomeric tau. And again, there was formation of nanoclusters. I also did some other controls on like expression level or method of expression, like stable versus transient. And I saw that in all cases, I got nanoclusters. So based on all the above data that I've showed you so far, we show that there is a population of tau that is both microtubule associated and oligomeric. So next, we wanted to look at our other cell line in the cell model, clone 4.1. So clone 4.1, I'm reminding you here, is the clone that contains cells where tau is very aggregated. And I have included here a representative image from this clone. So as you can see, it looks dramatically different from the images we looked at before, where tau was just on the microtubules. And you can see that different types of aggregates are forming. So I've zoomed into two different regions here just to show you that we get structures ranging from small nanoclusters in the magenta circles to short fibrils in the green circles to more branched fibrils in the yellow circles, and finally larger, more amorphous structures that are reminiscent of NFTs seen in patients in the white circles. So we wanted to, look at the nature of all these different tau aggregates, but we first started by focusing on the smaller nanoclusters that we have in this cell line, to compare them with the nanoclusters we saw in the non-aggregated conditions. So we performed the same fitting that I introduced to you earlier for the non-aggregated tau clone. And interestingly, here we got approximately the same number of monomers, so around 60%, but we seem to have much less dimers and trimers and more higher order oligomers than in clone 4 with the non-aggregated tau. So we thought that it could be that dimers and trimers are likely seeding the formation of higher order tau oligomers that consist of more than 3 tau proteins. And to further look into that idea, we performed again some two-color storm experiments. So same as for clone 4, we looked at GFP tau versus alpha tubulin and GFP tau versus oligomeric tau stained by the T22 antibody. And if you look at the colocalization analysis, immediately you can see that the very large NFT-like structures are in the isolated category, so they do not colocalize with tubulin, which makes sense, and also they do not colocalize with T22. That also makes sense because we didn't expect them to be oligomeric. What's interesting though is that we have some populations that are oligomeric and colocalized with tubulin. So we have approximately 17% of tau clusters that were microtubule associated. And if we compare the number of localizations per cluster in those clusters that are microtubule associated between the aggregated clone versus clone 4, which is the non-aggregated tau clone, you can see here in the plot, we see that there's a significant increase in the number of localizations per cluster. So basically, those microtubule associated clusters in clone 4.1 are bigger, which reinforces the idea I introduced before that we could have some seeding of aggregation on the microtubules before these structures dissociate to further aggregate. So next, we wanted to explore, the rest of the aggregates present in these cells. And for this step, we thought that it would be cool if we could distinguish the different tau aggregates based on their shape. So after much thought, we went into the development of an algorithm that performs shape classification on super resolution images, and it's based on unsupervised machine learning, and specifically a method known as Iterative Hierarchical clustering. So using this approach, we identified the presence of distinct tau aggregate classes. So I'm going to talk you through it a little bit. So the first stuff I'm going to show you is basically a proof of principle experiment that we did, proof of principle analysis that we did with the data. So what I did is I took super resolution images of clone 4.1, so aggregated tau cells, and after visual inspection, I divided them into two categories, those of low tau aggregation, meaning that they had smaller aggregates overall, and that's in green, and those of high tau aggregation, meaning that they had much larger aggregates, like imagine the NFT-like aggregates that I showed you. And then those images were segmented to represent, individual tau aggregates, and they were pooled together. So we had tau aggregates from both low tau aggregation and high tau aggregation. And then we ran the shape classification analysis. And it gave us 22 classes of different tau aggregate shapes. So we looked at the classes and we put them in categories. So for example, you see here tau fibrils, branch tau fibrils, and in particular, we plotted the results into these ellipse plots. So these ellipse plots are basically each ellipse corresponds to a different class. So you see in the first plot, we have classes 1, 2, 3, and 4. And the size of each ellipse corresponds to how many tau aggregates there are. So for example, like class 4 will have more tau aggregates than 1. And you cannot compare the sizes of the ellipses between the different plots, but you can compare them within the same plot. Also, the shape of the ellipse corresponds to the shape of the class that it represents. So more elongated ellipses correspond to more elongated shapes. And as you can see, the plots are area versus, excuse me, number of localizations. And we looked at structures that had over 500 localizations because we found that anything below 500 just simply didn't have much of a shape. So in the first three plots, we're progressing into bigger aggregate structures. We see that there's overlap between aggregates from low tau aggregation and high tau aggregation. And then as we move to even, bigger tau aggregates, we see that, for example, in the last plot, like the classes 20, 21, and 22, we mostly have representative aggregates from the high tau aggregation condition. So this worked, again, as a proof of principle that this algorithm can help us distinguish different tau shapes in a way that makes sense. So we wanted to specifically look at the nature of these tau aggregates. And what we chose to do was to look at some of the phosphorylation markers that tau can obtain. So as I mentioned, tau can undergo hyperphosphorylation. There are multiple different markers you can look at, but we chose to look at two of them. A marker that appears early in the tau aggregation pathway, known as threonine 231, and a marker that appears late in the pathway, known as serine 202, threonine 205. And basically, I stained with antibodies that recognize these markers. And I'm showing you here storm images, or representative storm images from the different stainings. And I've highlighted basically structures that are recognized by each one of those markers. And the idea behind this was that we were wondering whether they could be different tau aggregate species that have different markers. And just from initial inspection, visually we see that there seem to be more fibrils recognized by this late aggregation marker, and more of these branched tau fibrils from the early marker. So the next thing was to use the shape classification algorithm that I just introduced to you to perform shape classification and ask more questions about which tau aggregates are associated with which marker. So again, same as before, we pulled together tau aggregates from different categories. In this case, the categories were staining with the early phosphorylation, the early tau aggregation phosphorylation marker in magenta, with a late tau aggregation phosphorylation marker in green, and with GFP nanobody that is supposed to stain all tau aggregates in the sample in blue. And again, the result was that we got 23 different classes of tau shapes, which is very similar to the 22 we got before. And here again, I put them in categories like tau fibrils, branch tau fibrils, NFT-like aggregates, and so on. And when I looked at the numbers just to see if there's a specific staining that's overrepresented in a specific class, I found that, for example, the green marker, which corresponds to the late tau aggregation phosphorylation marker that's stained with antibody 88, was overrepresented in classes like class 5, which contains tau fibrils. Whereas the early tau aggregation phosphorylation marker, threonine 231, was overrepresented in classes that contain branch tau fibrils, such as classes 3 and 9. So, we thought that was really interesting, and we were wondering if, different tau aggregates could be, they could belong into like different tau strains, because this is an idea that's currently in the literature, that you could have like different neurodegenerative diseases stemming from different tau strains. We also found structures that didn't have either phosphorylation marker, which had us thinking that, okay, maybe there was some technical difficulty, like the epitope wasn't accessible, but it could also mean that there are structures that simply don't have any of these two markers that we looked at, and they could have different markers, again reinforcing the idea of like different strains, the possibility of having that. So we think that this algorithm has great potential in basically distinguishing different tau aggregates, and it could facilitate, the studies of that are looking at different tau strains. So to summarize this part, which again is all the published data, you can find it in our paper, I showed you that we found evidence that tau forms oligomeric complexes on microtubules, which are distinct from pathological aggregates. I also showed you that we have reason to believe there's some templating of aggregation on the microtubules prior to dissociation. And finally, that using a novel machine learning approach, we have distinguished different aggregate classes of tau based on their shape. So Now I want to move on to the second part of the talk, which is much more brief. And I mostly have very preliminary data. So, you know, anything could change. But I'm going to talk a little bit about the timing of tau aggregate clearance and, you know, how the different protein degradation pathways in the cell could be involved in this process. Sorry, I need to drink some water. So for this part, we again use the cell model that we previously used, we obtained from Virginia Lee's lab. And I should mention that in the first paper, this cell model appeared, they actually did look at tau aggregate clearance upon removal of doxycycline. So since the construct is DOX-inducible, if you remove doxycycline, you lose expression of soluble tau. So they saw that if you do that over time, you'll get aggregate clearance. And they mostly focused on what happens after day 5 without doxycycline. And they saw that autophagy is partially involved in this clearance. So we wanted to focus on sort of earlier stages of this clearance process because, you know, we have the tools to look at the smaller structures that may not be resolved with conventional light microscopy. So I started with a simple, oh yeah, well, before I tell you that, I just want to give you a brief introduction into the degradation pathways, and then I'll go into exactly what I did. So I'm going to start with like a textbook introduction of autophagy versus the ubiquitin proteasome system, the two main degradation pathways in cells. So normally when we think of autophagy, autophagy is known to degrade long-lived proteins or protein aggregates or damaged organelles. And this process starts with formation of the phagophore that encloses those damaged proteins that then fuses into the autophagosome, which eventually fuses into the lysosome, where the hydrolases will degrade the damaged proteins. Now, the UPS is traditionally thought to degrade short-lived proteins that are smaller in size and can fit through the proteasome core. So these proteins, as I said, they're small, like sometimes monomeric, and they are marked with a ubiquitin chain, which acts as the degradation signal, and they are unfolded inside the proteasome and degraded. Now, tau can actually be degraded by both pathways. Traditionally, it's thought to be an ideal substrate for UPS degradation because it can exist as a monomer, it's naturally unfolded, and can fit through the proteasome. And tau aggregates, simply because of their size, they're thought to be degraded by autophagy. The current gap in our knowledge, though, is that there is evidence that tau aggregates can also be associated with the proteasome, but we don't know if they're actually degraded. Sometimes they're inhibiting the function of the proteasome. And the main gap is that we don't know, in terms of tau aggregates, which tau aggregates get degraded by which pathway, or if they can be degraded at all. So there are many unanswered questions that we have, and things can become further complicated because the two pathways, there's an interplay between the two pathways in some cases. So that's why we wanted to use the cell model, which is sort of a simpler system to look at tau aggregation and clearance, and if possible, specifically focus on the clearance of certain tau aggregates. So the first thing I did was, similarly as what they did in the paper, I looked at tau aggregate clearance after the removal of DOX. So I removed DOX for 1 day, 2 days, 3 days, and 5 days in cells, and then I performed storm imaging. And I'm showing you here some representative images from day 1, 3, and 5 of DOX. So as you can appreciate, for example, in the first image, we still have, some of these very large NFT-like aggregates. In day 3, not so much. And in day 5, in this case, we are left with many fibrils. As you can see from the plot, the plot shows the total localization density, for the tau in all of these days. And you can see that as we progress into, for example, day 5 without DOCs, we have much less dense structures. And that's indicative of aggregate clearance. Then we use the shape classification algorithm that I introduced to you earlier, and we wanted to basically confirm what we see visually with this shape analysis and see if we could tell if there's a priority in the clearance of certain structures. So here I combined images from day 1, day 2, and day 3 without DOCs in blue, green, and brown respectively. And as you can see, we do have an overlap in terms of structures, but what's interesting is as we progress into structures that have higher number of localizations, we end up with more tau aggregates from day 1, which is, we still see those NFT-like structures. And as we move into day 2 and day 3, we have less and less of these larger tau aggregate classes. So we see that there is a priority in clearance of these large tau aggregates. So we're hoping to use this algorithm for further characterization of the rest of the shapes that are present and sort of what comes next. So I'm currently trying to address the question of which clearance pathway or pathways are regulating this clearance. So I'm trying to treat cells with different inhibitors of the proteasome, autophagy, as well as combining both proteasome and autophagy inhibitors. Again, I only have very preliminary data, but I'm showing you some data from proteasome inhibition experiments that I've done. So in particular, I used an irreversible proteasome inhibitor called epoxamycin. And what I did here is I focused on days 1 and 2 without DOCs. And I'm showing you some representative images of the epoxamycin treatment that I performed. So if you look at the plot, I'm showing data. Again, we're looking at densities from day 0 and day 2 without DOCs, and we see a drop in the density, which makes sense, as we saw before, because we have clearance happening. But if you look at the third plot, which is basically 2 days without DOCs, where the second day I also added epoxamycin, so 1 day treatment with epoxamycin, you see that we have a very large increase in the density, which we think corresponds to accumulation of structures because the proteasome is inhibited. So this gives us an indication that we could have involvement of the proteasome in those early days of DOCs removal. And then when we're looking at the last plot on the right, I've treated 2 days without DOCs and 2 days with the epoxamycin proteasome inhibitor. And we see that, again, we have an increase in density if we compare it to day 2 without DOCs, but we have a decrease if we compare it to 1 day of treatment with epoxamycin. And we think that it could be possible that there's compensation of clearance from autophagy, but of course, I don't have evidence right now to know that. But this is sort of a next step I'm trying to explore. So I know this was very brief, but the next steps for this project are, as I alluded to it earlier, look into the timing of tau aggregate clearance. And specifically, we want to know if there are certain tau aggregate species that seem to persist, like they're more resistant to this clearance. And we sort of have a feeling that it could be that fibrils are more resistant. And we want to know, if possible, like which tau aggregate species, if there's a preference in terms of which pathway degrades which specific tau aggregate species. So thank you so much for your attention. I would like to thank again the Center for Engineering Mechanobiology for putting together this seminar series and for giving me the opportunity to present. This was truly an honor, and I'm very excited to hear the next talks coming up. I would really like to thank Melina, my mentor. She is amazing. I look up to her and admire her so much. Thank you, Melina, for all your help and guidance. I have also highlighted other people that are very important, that were very important for the published work that I showed you. Aryan really helped with the analysis and also developed the shape classification algorithm that I presented to you. Thank you so much, Aryan. Adriana helped very much in all of the phosphorylation marker imaging experiments. Thank you, Adriana. Elena was really helpful as well in experiments and reagent preparation. Thank you, Elena. And I would also like to thank Virginia Lee for getting us, giving us the cells for the cell model, Chris and Gia for the Ddc algorithm, Peter for the colocalization algorithm, and Chantel from Erica Holzbauer's lab for helping me with the neuron preparation, as well as my thesis committee and all of our funding sources. So thank you again.
Thank you, Melina. Very nice work.
Thank you.
All right, while the questions are being put here in the chat.
Should I stop sharing, or I don't know if I can see them.
It's up to you. They may want to see, maybe they want to go back
Okay.
into some of these images. Let's just see. But
Let me see if
while
I
the
can
questions
see
are
the
good.
chat. Oh, I can see the chat.
Yeah, so here we go. I don't know if Vivek is on. I'm not sure what images he's referring to. So his question is, can you tell where the MTS are in this image?
Okay, sorry, I wasn't able to see the questions as I went in the talk. Which image is that for?
Yeah, please ignore that question because the very next slide had the answer.
Okay. All right.
Okay, thank you.
I have a different question. So, one of the roles, tau is a microtubule stabilizing protein, but it's also a microtubule cross-linking protein, right? And particularly in brain injury and things, the cross-linking is important. So my question to you is, from your images, can you tell if microtubules are being cross-linked by tau?
So there is evidence of bundling of microtubules, and that was sort of our, like before we did any of this characterization, we wanted to know, because the cells we use normally don't have endogenous tau. So we wanted to see, when you engineer the cells and you add tau, we know that it's not the same as neurons, but we wanted to see if tau has some of its normal functions, and it does bundle microtubules, if that's what you're referring to. So I would say yes. So it does perform some of its functions that it does in other cells.
All right, thanks.
Sure.
Okay, the next question is from Kevin Foskett.
Sure.
What does tau do, and does it make sense that they would be oligomeric to accomplish this?
Yeah, so tau is, important for stabilizing microtubules and promoting polymerization of microtubules. We do think it makes sense, and there's actually evidence from in vitro data that, they've put like tubulin and tau together, and they saw that like tubulin can help the oligomerization of tau protein. We haven't looked specifically at function, so I can't say that it, I don't know what exactly they're doing based on, the data that I've obtained. But I think it's not like there is evidence that from in vitro experiments that, you can have oligomer formation, along with tubulin. And there's also the idea of like liquid condensates forming on microtubules, liquid condensates of tau. We don't really think that what we see is liquid condensates just because it's much smaller than the condensates they have seen in the literature. So yeah, I think that's what I can comment on so far. So I would say it does make sense, but what exactly it does based on our imaging, I don't know.
Okay, the next question is from Stacy Rakowecki. Maybe saying that wrong, I'm sorry. Okay, beautiful imaging. Were you able to see differences in microtubule structure in the pathological state when fewer clusters were, I guess, microtubule associated? In other words, does the atypical distribution of tau affect microtubule structure?
Yeah, that's an excellent question. Yes, we actually looked at tubulin density, and we saw that tubulin density goes down when tau is microtubules. So we do believe that the aggregates have an adverse effect on the microtubule structure, even though the cells are viable. So it's not like to the point where, you know, they're dying or anything, but it does have an effect when we compare it to the clone 4, where tau is on the microtubules. And also we compared to cases where we just didn't express the tau construct and looked at the tubulin. So yes, to that.
Okay. Next question is Charlie Anderson. Is the shape classification algorithm working in 3D, or is it analyzing 2D images?
Yeah, great question. So everything, all of the imaging I showed you is in 2D images. So the algorithm is working in 2D images. But I think, so I think it can work. Like, I've never done 3D imaging, but I think it possibly could. Adriana, have you guys used that on your 3D images? Adriana is another grad student in the lab that has done some images. You haven't used it yet, or?
Yes, hi, I'm Adriana. So just to give a quick background, I'm imaging tau aggregates, but in a tissue context. So I'm using Alzheimer's disease patient tissue brain. So we did try to do some 3D star images of these tau aggregates to see, to get more details of the morphological features that these tau aggregates contain in the disease and in tissue context. Unfortunately, the algorithm itself is not capable yet of analyzing these structures at a 3D, but it is possible.
That's what Thank you, Adriana, for confirming. Yeah, but I think it's possible. Like, we're trying to sort of use it in many different ways. And we've already, like, I say we, but Aryan has been doing all of this optimizing, like, since, you know, we use it for the paper. There are more features that have been added to it. So I think eventually it could work on 3D images. So, yeah.
I think that was actually Michael, well, related question. Maybe that's the challenge. So you worked with cell lines, I guess, with patient cells or maybe harvested brain sections. Is this the challenge potentially with getting images, if you can get images?
Yeah, so as you said, I worked with cell lines. That's sort of a much simpler thing. You know, they're easier to culture and all that. And I'm sure, as Adriana can confirm, like, tissues, just even like preparing the tissue for storm imaging is really challenging itself. Like, that needs, you know, it took Adriana like, you know, months to fully optimize for imaging with storm. So that's, I think, the next exciting step for what we've showed to, you know, do some of these characterizations on tissues from patients. So yeah.
Good. All right, any other questions? I think that's it that I see. All right, Melina, thank you very much for your exciting talk.
Thank you so much.
And again, I would just like to announce again that the Center for Engineering Mechanobiology will have this seminar series monthly, and the next seminar is, let me just see, Annie, maybe jump in, hold a second, is June 16th. And that is, you'll see the announcements. So this is Saeed Musavi would be the next talk. So that's plant mechanotransduction, the role of piezo ion channel in sensing external forces in plants. So that's on June 16th. All right, thank you very much, everyone.
Thank you. Bye.